We'll just finish up here with the last part of Chapter 6, so your test obviously will go through Chapter 6. So now, as far as preserving bacterial cultures, there are several ways you can, once you've cultured out a particular bacteria, there are several ways you can save it. Deep freezing basically is where a pure culture of microbes is basically placed in a suspending liquid, quick frozen at extremely low temperatures. You can freeze dry lyophilization, basically whereas the suspension of microbe is basically quickly frozen at a temperature at about 54 degrees to 72 degrees. And then the water is basically removed by a high vacuum. This is basically freeze drying. This is used quite frequently in, especially in labs that preserve different types of bacterial and or viral elements for prolonged periods of time. The remaining powder can be stored for many years. Surviving microorganisms basically are cultured by rehydrating them with a suitable liquid nutrient. Now this obviously wouldn't work for every microorganism because some of them will not survive this freeze drying process, but there are many microorganisms that can survive this process. The reproduction of more primitive cells, such as the prokaryotes, you can use a growth of a bacterial culture. You can also see in the growth phase they produce by different methods: binary fission, budding, conidiospores, and fragmentation filaments are various ways that we see reproduction take place in more primitive prokaryotic cells. Your book gives some illustrations of binary fission. Bacteria normally produced by this method, genetic materials basically become evenly distributed. Then in a transverse walls, they form across the center of the cell, and then it separates in two cells. You basically will see a little furrow develop between the cell, and then two individual cells will be produced. A few bacterial species use the budding process. That is, the initial outgrowth enlarges the cell size, and then it will separate. And then some filamentous species produce reproductive spores or simply fragment into viable pieces. This is just an illustration out of your book demonstrating the binary fission. Again, you see the cell elongates, the plasma membrane will divide, cross walls will develop, and two separate cells are generated. This is just an electron microscopy of this mechanism taking place in a particular type of microorganism. Now the generation time, you need to know this definition. Generation time is the time required for a cell to divide or a population to double. That's referred to as generation or doubling time. Bacterial populations are usually grafted logarithmically rather than arithmetically to produce the handling of immense differences in the numbers. Now this is important because bacteria typically will grow logarithmically. It means it will grow extremely fast, and so many bacterial infections that you see, the population today has grossly increased in a 24-hour period. That's why culture medians are a very effective way to be able to evaluate certain bacteria, because they multiply so quickly. They multiply so quickly that initially when you inoculate a culture median, you don't see the bacteria, but yet within 24 hours you see significant colonies, which is reflective of heavy bacterial growth on the culture median. When bacterial populations changes and the bacterial growth is basically charted or plotted, it becomes very apparent what the significant growth phase is. There are two phases you should be familiar with. The lag phase, which shows little or no cell division. However, metabolic activity may be intense. And remember, a lot of times before you see cell division take place, even in the human cells, there is an increase in the production of internal structures. You may see an increase in mitochondria. You may see an increase in the amount of protein synthesis that takes place. So ribosomes that attach to endoplasmic reticulum, you see an increase in the production of protein activity in these cells, but you may not see division. And so this is referred to as a lag phase. In the log phase, the cells are reproducing most actively, and the generation time actually reaches a minimum and remains constant. A logarithmic plot actually produces an ascending straight line, and I think I have an illustration for that coming up. They are then the most active metabolically and most sensitive to adverse conditions. In a chemostat, It's possible to keep a population of such exponential growth indefinitely. Without a chemostat, however, most microbial death will start occurring, basically because eventually the balance of the number of new cells is reached and a stationary stage is reached. Now that can be dependent on a number of things. The amount of nutrients that are available to support the bacterial growth, because remember, all of these bacteria are competing for the same nutrient source. So at some point there will be a plateau or a stationary phase, but in a chemostat they can keep generating exponential growth indefinitely. When the number of deaths exceed the number of new cells formed, that's referred to as the growth phase, and then in a logarithmic decline is reached when significant amounts of nutrients have been produced. Also, one thing to consider, certain bacteria, there are byproducts that are produced by the bacteria, or waste products. And once in an isolated medium, if too many waste products are produced, the other cells or the cell itself can't survive if it's locked into that. And that's the advantage of using a chemostat to be able to identify numbers. This illustration is out of your book. Table just kind of shows you the number of cells that can be generated in a short period of time. If we looked at the actual growth phase, you can see the number gets up in the millions. But if you use it on a log scale, it's much easier to report. So typically, when we talk about bacterial growth and we refer to the log phase, you're typically just using a number to be able to identify a quantity. But remember, it's a logarithmic number that's being reported. 100 cells growing in 5 hours produced over 1 million cells, close to 2 million, and basically easier to report this on a logarithmic scale. Should be familiar with this graph. This demonstrates to you the lag phase. Again, in the lag phase, it doesn't mean that the cells are dying. It means that the cells are increasing the intracellular metabolism, catabolism that takes place to go through the reproduction. And then in the log phase, you'll see significant growth. And then there's a stationary phase, and either dependent upon changes in the environment, nutrient supply, or whatever, death is basically a logarithmic supply as well. Plate counts basically refer to dilutions of bacteria suspensions that are distributed into suitable solid nutrients. Medians by serial dilution, and colonies appearing on bacterial plates are counted this way. Colonies do not always arise from single cells, but from chains or clumps, or counts are often reported in colony-forming units. And then plate counts, you inoculate a petri dish, basically inoculate, adding the bacteria to the plate. And basically, as you add it, the melted nutrients swirl into a mix. Colonies will grow in this solidified medium. Then you can spread it in the plate as well, and again, colonies will grow on the surface of the medium. There are also ways to do plate counts. Actually, what laboratory technicians do, if I can get it out, what they basically do now for colony counts, they can do serial dilutions to be able to identify organisms. This may seem like a good culture because it's got tons of bacteria on it, but sometimes it's hard to differentiate, and so the best thing to do is to serially dilute, so you got one targeted area that you can take out a specific bacterium and be able to look at it. There are some other methods how you can measure the number of microorganisms that are grown. This is called a filtration method, where bacteria can basically be sieved out of a liquid suspension onto a thin membrane, filtered with a pour to 2 small bacteria pass through. This filter can be transferred to a pad soaked in nutrients and medians to be able to be determined. This is a filtration method, and again, sometimes colonies are reported per high-powered field, and they'll take a grid, and you see how this culture plate has been gridded, and they'll count the number of microorganisms in a particular grid, and then there's a formula that number is inserted into, which gives the actual count. And that's where you come with the term most probable number. Most probable number method. Sample is basically diluted in a series of tubes or liquids. The greater the number of bacteria, the more dilutions that it takes to dilute them out entirely and leave the tube without growth. Results in such dilutions can be compared, statistical tables, and cell counts can be estimated. And this is a serial dilution mechanism that can be used. This is a direct method where basically you count the positive tubes and you compare it to a table that's used to give you an estimate of the number of organisms. The direct microscopic count is basically you measured a volume of bacterial suspension. It's placed in a defined area on a special microscopic slide, which have several designs, and then the microscope is actually used to count the number of cells in a microscopic or a high-powered field. The average number by field is then multiplied by a factor or formula, and that's what gives you an estimate in the number. And these illustrations are out of your book and demonstrate that. The last concept that I wanted to talk about is just turbidity. You can estimate the turbidity by shining a beam of light through a solution. A bacterial suspension can demonstrate turbidity based upon its ability to allow light to pass through. This recorded or absorption is called optimal density, and a spectrometer is used to determine the number of bacteria. As you can see in the illustration, light can pass through this tube because there's no turbidity, and the spectrometer gives a particular number. If there's a significant amount of bacteria in the solution, then minimal amount of light is passed through. The spectrometer only gives a certain reason, and you can get a relative bacterial count based upon that spectrometry. Okay, and I'll stop right there. And again, your exam will go through this portion of Chapter 6, and again, the review session will be set up for 4:15 on Sunday. We'll meet down in the lobby, open up the door for you there. I'll also try to have the review on the media for you as well before that time, so that way those of you who